What I'm going to present today is some work on finding groups of genes with alterations that are associated with functional association in cancer. And just to, let's start introducing some concepts that have been explained yesterday by Quaid and Ben anyway. In particular, we're going to look at somatic mutations, since cancer is a process of accumulations of mutations, in which we start from a normal cell, that accumulating mutations, most of which are random passenger mutations that do not have an effect on the cell, leads to a population of cells in which we have these passenger mutations and also some driver mutations that confer selective advantage to the cell. Okay, so what we can do is take now the population of cells and do bulk sequencing, so obtain the collections of mutations observed in a cancer patient. And of course, when we sequence a cancer patient, we do not observe which mutations are drivers and which ones are passengers, but we only see that there are somatic alterations in this patient or maybe in the genes of these patients. And for the purpose of this talk, when I talk about alterations, I mean both single nucleotide variants, so changes at the single base, or longer and larger alterations like deletions or amplifications of in bigger portions of the DNA. So, recent studies, like the studies from TCGA or the International Cancer Genome Consortium, have analyzed a large number of tumors. So I guess the latest release from the ICGC now gives access to more than 20,000 samples from tumors, and using these tumors we can try to understand mutations in cancer. And in particular, as I guess Quaid said yesterday, what we have understood by analyzing this data is that, or what we think about cancer genomes, and something that we have, some insights we have gotten from analyzing these genomes, is that there is a small number of driver mutations in cancer, some number between, let's say, 2 and 10 or 2 and 20, something like that. And there are many more actually passenger mutations, hundreds up to tens of thousands of passenger mutations in a typical tumor. Okay, so the main challenge that we want to try to solve by looking at all this data is try to identify the mutations that are related to the disease. So driver mutations or anyway mutations that have an impact on the tumor. Okay, so the common approach to look for these driver mutations is to look for recurrently mutated genes. So looking at all the samples from a given cancer type to consider one gene at a time, and then look, for example, at a simple score for the gene that could be the frequency of mutation of the gene, so the number of samples in which the gene is mutated, compare it with what you would expect if the gene didn't contain any driver mutation, and then look at the significance scores for all the gene, and by correcting for multiple hypothesis testing, you identify a small number of genes that are significantly mutated, so they have a significant score. And for example, this is a figure taken from a study from the Cancer Genome Atlas. And these are the genes that are reported as significantly mutated in that study. And so we can already observe, when you look at these genes one at a time, so in isolation, what you report are genes that are frequently mutated in cancer. So the genes that are identified as driver genes by this approach are genes that have a relatively high frequency of mutation in the cancer patients. And there is then this long tail of genes that are not reported as driver genes containing driver mutations, but still are probably, at least some of them are going to be important for cancer, and this is something that we can understand also by the fact that many patients do not contain any alteration in the gene that we report as driver genes by this simple test. And just to understand why this simple test may not work perfectly, this is due to the fact that there are also many confounders that one has to take into account when we have to try to estimate the background mutation rate for the different genes, since the probability of mutation of a gene depends on many factors, like its length, the expression level of the gene, the replication timing, and many more other factors that probably we don't even know all of them. So in trying to then to identify the mutations that are important for cancer, but that are not frequent enough to be identified by this simple gene test, many approaches have relied on the idea that cancer is actually a disease of pathways. And this means that actually the cell is a complex circuitry of interacting proteins and the interacting genes, the corresponding interacting genes. And basically one can, by simplifying a lot, one can think that there are different mechanisms in this circuitry, different subnetworks in this circuitry that needs to be altered in order to develop cancer. And again, simplifying, one can think that there are different subnetworks corresponding to the different hallmarks of cancer that need to be acquired in order for cancer to develop. And the fact that now an entire network is associated with a given trait of cancer also explains why there may be some genes that are mutated with very high frequency because they are part of a network and they are the commonly mutated gene in the network, while there are many other genes that are mutated with smaller frequency but have the same effect on the cell. Okay, so instead of looking at single genes, so at genes in isolation, a better way to identify driver mutations or mutations important for cancer would be to look at pathways, so at these sets of interacting genes, and try to identify which ones are significantly mutated. So the first approach in trying to do this is to look, for example, at known pathways, so subnetworks that are known to perform some function, and by generalizing the test for single genes, one can identify some subnetworks that are mutated in large number of patients. However, this approach that looks at known pathways has several problems. For example, not all pathways are known. There is crosstalk between pathways. They are not separated entities. And sometimes these pathways that you find in databases are so large that when only a small portion of the pathway is important for cancer, you are not actually able to detect the pathway is significantly mutated. Okay, so what something I've worked on a lot in the past years is how to try to find these mutated sets of genes, this pathway, without restricting to known pathways. So without relying on the information in databases that tell you what are the pathways and what is their function. So the problem when you move from using known pathways to not relying on this type of information is there are many candidate sets. So for example, if you were to screen all possible subsets of at most 6 genes, there are more than 10 to the 20 sets, and this poses a problem both computationally and statistically. However, there have been many methods that have been developed to identify these driver pathways and these interacting set of genes that are important for cancer, some of which I've contributed to. And there are many more methods that are not shown in this slide. What I want to focus on today are methods that do not use only genomic information to identify these driver pathways, but also try to leverage on other data that is sometimes available from these larger studies. In particular, either clinical data or molecular phenotypes. And in particular for today, I'm going to focus on molecular phenotypes. And so what we're going to see today are 2 algorithms that try to use these phenotypes, and I'm going to try to give you an idea what are the phenotypes that we looked at, and try to identify these important set of genes for cancer using both genomic data and this phenotype data. Okay, so the 2 algorithms I'm going to describe are one is called Uncover and finds groups of genes that are associated with a functional target measure. For example, you can think of it as a measure, a real value that tells you something about, for example, drug response or drug sensitivity for your sample. And the second algorithm is Netflix that looks for subnetworks associated, again, with the target measure. Okay, so let's start from Uncover. And Uncover builds on the idea that in many cancer pathways, usually only one gene in the pathways is mutated in each patient. Okay, so if you look at a cancer pathway, like it could be this one, for example, if now you look at all the patients that have a mutation in this pathway for a given cancer type, then for most patients, only one of the genes in the pathway is mutated. Okay, so while there are many reasons why mutual exclusivity could arise, it provides us with a signal that we can use in trying to identify these driver pathways, so these sets of interacting genes that are important for cancer. And building on this idea, there have been many methods that have been developed by the community, and these are, I think, all the methods up to 2017. There are others that have been developed after that time. Okay. So with Uncover, what we want to do is then to use both mutual exclusivity and the target profile to try to identify these pathways that are associated with the target profile. So just to give you intuitively, the first thing you could try to do is then to say, well, instead of using the target profile, I could just try to identify subsets of genes that show high mutual exclusivity in the cancer cohort, and then see if these set of mutually exclusive genes is actually associated with the target profile. Okay, so kind of a post-processing to find the subsets of mutually exclusive genes that are associated with the profile. Okay. However, if you try to do this, actually, you do not identify all the subsets of mutually exclusive genes that have an association with your profile. And the main reason is that there are mechanisms that are altered across all the samples that you have, and this strong signal actually masks the signal that you are interested in. So what we want to do is really take these mutation data, these alteration data, in which for each sample we know which alterations are present in the sample and the target profile, and try to identify this set of mutually exclusive genes such that in most patients only one of the genes in the set is altered, and there is a preferential alteration for one of the two ends of the target. So for example, here in orange, we have a set of 3 genes that are associated with a high target value for the samples. Okay, with the idea that if, for example, we are looking at target profiles that describe drug response, these set of genes may correspond to an underlying mechanism that is driving the response to the drug. Okay? And that means that in the set we may or may not have the drug target, but we also we can identify other genes that are important in predicting the response to our drug. Okay, so our goal is to find this mutually exclusive set of alterations associated with the target profile. So more formally, to see what's the problem definition, we are given this target profile, a set of samples corresponding either to cancer patients or, for the data we will look later on, to cancer cell lines. And we have also a set of genes such that for each of these genes actually we know in which samples they are mutated. So just to, without loss of generalization, let's assume that our samples are sorted by decreasing value of the target, and the target is, again, real value could be positive or negative. For each of the genes, we know in which set of samples it is altered. For example, this green gene here is altering these 3 samples. The yellow gene here is altering these 2 samples, and so on and so forth. And then starting from this data, we actually associate to each sample a penalty that can be, for example, the average value of the target, or there are different ways to set it. So for each sample now we have a weight and a penalty. And our goal is to identify a set of k genes such that the sum of the weights of the elements that are covered by the set is maximized. So we have a high association with the target value. And there is a low overlap between the selected sets. So this corresponds to enforcing mutual exclusivity between alterations. Okay, so slightly more formally, given the set of samples and the alterations for each genes, the weights for each sample and the penalty, what we want is a set of at most k elements, set of at most k genes that maximizes the following weight. So the weight is given by the sum of the weights of the samples for which we have at least one alteration in the sample, minus the penalty that is given by the number of times that the sample is covered in excess to only once, times the penalty for that sample. Okay, so now this gives us a well-formulated computational problem. We can study it and try to propose algorithms for it. And so the first results actually that we have is that this problem that we call the target-associated k-set problem is computationally difficult, so probably there is no polynomial time algorithm to solve all possible instances, but we still want to solve this problem. And so we first proposed and studied the greedy algorithm to solve the problem. That is a natural choice since this is a kind of a weighted coverage problem, and the greedy algorithms have shown to perform well for this type of problems. So the greedy algorithm is fairly simple. We start by computing the total weight of each set. So for each gene, we computed the weight of the patients in which the gene is altered. So for the gene that is altering these 3 patients, we compute his weight. For the genes altering these 2 patients, these 2 samples, we compute the weights, and so on and so forth. In the second step, we just select the gene that maximum positive weight, and in this case, it will be the first gene. Then we update the weights so that instead of the weights for the samples that we just selected by taking the corresponding genes, we now, instead of the weight, have the corresponding penalty. Okay? And now, by repeating this process up to k times, we're going to select in total k genes. So maybe in the next iteration, once we update the weight for the second gene, its weight is going to be minus P3 plus it's the weight for the fourth sample, and we select the gene, again, we put it in our solution. At the end of this process, we report all the genes that we found. Okay. And in this way, we can report exactly k genes or report at most k genes if we stop whenever we cannot improve the score of our solution. Okay, so this is a greedy algorithm, it's the natural greedy algorithm for the problem. And so actually we first provided theoretical analysis for the algorithm showing that it returns an approximate solution with some guarantees, and in particular the score of the solution you report is at most a factor 1 over k far from the optimal solution. And the bad part of these results is actually there are instances for which this bound is tight. That means we don't provide a great approximation, in particular if k is large. But this is a type of a worst case analysis. And so what we did instead was also do a type of a probabilistic analysis in which if you assume that the data that you get is not adversarial but comes from a generative model in which a set of genes is associated with your target and the other mutations are essentially random, then we can prove that if you have enough data, your greedy algorithm will really find the set of genes associated with the phenotype. Okay, so this is for the greedy algorithm. And we then also proposed an integer linear program to solve the problem. And the integer linear problem is fairly simple. That also helps to have a fast algorithm in practice. And so just to give you an idea of the details of this integer linear program, we are trying to maximize, this is just a different way to write the score for a solution. So we try to maximize the cover for samples of high weight, and then we also, in our objective function, we subtract the penalty, the sum of the penalties for the samples, weighted by the number of times in which we cover each sample. This constraint then tells us that we want at most k genes in our solution, and then there is a set of auxiliary constraints to make sure that what we report is actually a valid solution. Okay, so we implemented our algorithms, both the greedy algorithm and the ILP, in our tool Uncover. Then it takes this alteration information for large number of samples and corresponding profile for each sample, where we have a value for each sample. And by running either the ILP or the greedy, we obtain sets that are set of alterations that are fairly mutually exclusive and associated with the target profile. It could be either high value of the target profile or low values of the target profile. We also perform a permutation test, which we keep the alteration data fixed and we permute the target profiles. And in this way, we can obtain a p-value that tells us how much of what we observe is actually due to the structure of the alterations compared to the association with the target. Okay, so the code is available at this link, and this is the work that is being done in collaboration with Rebecca Sarto-Basso, that is a PhD student at Berkeley in the Ockbaum lab, and by myself. Okay, so let's see now at some results. The first thing we did was to try our method on simulated data with the goal to understand mostly how well our algorithm can identify a set of alterations that is truly associated with the target, and the independence on the number of samples that we have. So in particular, the data we simulated in the following way. We planted a set of 5 alterations with association with the target, and we varied the strength of the association with the target, and all other alterations are placed independently of the target. And then we run the greedy algorithm or the ILP, and we report here in this figure the fraction of the genes in the set associated with the target, so the solution we are looking for, that is identified by our algorithm and this is the average value across 10 different randomized data sets. And we look at how this fraction varies with the number of samples that we have. Okay. As we can see, when the number of samples is fairly low, around 200, we can identify the set associated with the target only when there is a strong association between the set of genes and the target. But when the number of samples increases, then what we have is that both the greedy and the ILP improve in their performance. And in particular, the ILP does a very good job in identifying the true set, the set we are looking for, when the number of samples is around, let's say, 1,000. Okay. So this is encouraging since the sample size that we have now is usually less than 1,000. It may be around, let's say, 500, as we will see later on. So we also evaluated the running time of our method, since we are interested in running this algorithm on large-scale studies where there are many different targets that have been measured for the different samples. And what we have is that the greedy algorithm is very efficient. It takes only, let's say a few seconds to run, even for 1,000 samples. The ILP is not as efficient, as fast, but it's still fast enough that we can actually use it for large-scale studies. Okay, then we look at real data, and in particular we looked at drug sensitivity data given by the cell survival assays and provided by the Genomics of drug sensitivity in cancer. And we have this data for 265 compounds, so we have 265 different targets. And the alterations come from the Cancer Cell Line Encyclopedia, that provides the alteration data for more than 700 cell lines. So the first thing that we looked at is the impact of using the target compared to not using it in finding the set of mutually exclusive genes. And here on the left, we have for one specific target the set of 3 genes that is reported if you don't consider the target. And as you can see, these are mutually exclusive sets. And just to explain a little more on the figure, the top row is the target profile. And then the next 3 rows correspond to the 3 genes that are reported in output, so the solution. And the last one is the summary, that is essentially the OR of the alterations. So there is a black rectangle here if there is at least one of the genes in the solution that is altered for that sample. And the columns correspond to the different samples. Okay, so if we don't use target values, we report a set of mutually exclusive genes that cover and are altered in all possible samples, essentially, or in most of the samples. Instead, when you consider the target, and you look, for example, with an association with the low target values, you identify a set of genes that, again, are mutually exclusive, but there is a clear tendency of having alterations only in for one of the 2 sides of the values of the target. So, for example, these are for negative values of the target. And the same happens when you consider the other tail for the target. So this is the set of genes reported when you look for association with high values of the target. Okay, so and this is something that happens not only for few targets, but is usually the case. So in general, there are only 11 solutions such that the solution without using the target compared to the solution when you do use the target share at most 1 gene. You never share more than 1 gene, so the target actually has a huge impact on the results that you obtain, as one would expect. Okay, so now let's look at some specific cases and results that we obtained. And we looked at, as a case study, at the results that we have when we look at the sensitivity profile to a MEK-inhibitor. And again, the alterations are given from the Cancer Cell Line Encyclopedia. And again, the number of cell lines that we have here are around 490, around 500. So the numbers that I mentioned earlier. So again, this is the profile, and these are the alterations that are identified by our method Uncover. And as we can see, the alterations are essentially in genes NRAS, BRAF, and KRAS. And these are 3 well-known genes that are oncogenic activator of the MAPK signaling. And this is makes a lot of sense since MEK is a member of the same signaling pathway, and it is activated by mutation in the MAPK pathway. So we automatically identify a set of interacting genes that are part of the same pathway of the target of this inhibitor. Okay, so this gives us some hope that what we are looking for is actually biologically meaningful. And what we did next, before getting to larger scale studies, was to compare our method with the only method that looked for a set of mutually exclusive genes and that could scale somehow for large studies, that is the method Revealer. Okay, so what this table is showing is for 4 different targets that were used in the Revealer paper. The solutions that are reported by our method Uncover, in particular using the ILP, the solutions obtained using a Revealer, and we report here the objective value of our problem, the IC score that corresponds to the information coefficients, that is essentially the score the weight that is used by a revealer in trying to identify this set of genes, and the running time of the two methods. And there are some interesting observations. In particular, if we look at the IC score that is used by a revealer to identify this set of genes, but not by uncover, what we have is that in 2 cases out of 4, actually, the solutions we report have a higher IC score than revealer. And again, our algorithm doesn't consider the IC score, so this is fairly surprising, and we are essentially doing a better job than revealer in identifying solutions according to the score used by a revealer. And the second interesting observation is that if you look now at the running time, our method only takes, let's say, most 40 seconds for these 4 datasets, while revealer takes up more than 1 hour for one of these datasets. So our method is much more efficient and can really be used for larger studies. So we also used our method and compared it with the revealer on these GDSC drug sensitivity datasets for 265 compounds. We took a lot of time to run revealer, so it took a few weeks to actually get the results for these experiments. And we actually did a comparison only for one side of the target, so I guess for positive association with the target. And the main highlights of this comparison is that, again, if you look at the IC score used by revealer but not by uncover, the solutions reported by uncover are better than revealer according to the IC score in more than half of the cases. And if you then take another measure of correlation between the alteration profile and the target that is given by the biserial coefficient, point biserial coefficient, that is not considered by the two methods, in 95% of the cases, the solutions we report are better than the solutions from revealer. We also look at the some statistics in terms of enrichment for cancer genes of the solutions reported by the two methods. And what we have is that the solutions by our method uncover are much better in enrichment, both for cancer genes than for pathways than the solution reported by revealer. So this shows that, at least for this dataset, uncover is reporting a lot of interesting genes and a lot of interesting subnetworks. Okay, and in these slides, I just want to show 2 specific cases reported by revealer. So the first one is the subnetwork of 3 genes that again corresponds to part of the MEK signaling pathway that consists of the genes BRAF, KRAS, and NRAS, in particular single nucleotide mutations, that we obtain when we look at the compound VX11E, whose target is the RK2, that is actually part of this MEK pathway, so this makes a lot of sense from the biology. In some other cases, actually, what we obtain is maybe not as clear the biological significance of what we see. For example, for sunitinib, we obtain a set of 3 genes that contains RB1, a well-known cancer gene, SETD2 that is somehow associated with the protein localization to chromatin. And so there may be some relation to the sunitinib, whose target is the RTK pathway. But in this case, the solution is not great, I would say, from the point of view of the biology. So since we had good results with uncover, but not great results, I would say, then we tried to improve on it. And the way we improved was in developing a new method called the NetFix that now takes into account not only alteration data and the target data, but also interaction data, and looks for subnetworks of a large interaction network in trying to identify this set of genes that are associated with the target. And this is a natural direction since we're really looking for a set of interacting genes and proteins. Okay, so what the problem NetFix tries to solve from the abstract point of view is given this drug response profile and the alteration data for different samples. Now we are looking for a subnetwork, so a set of genes that is connected in an input graph, and that shows alterations that are approximately mutually exclusive and that they show an association, for example, with decreased values of the target. Okay, so again, we can formulate this problem more formally as the phenotype associated connected coverage problem. So we're given an input to the graph where the vertices are the genes and edge means that there is an interaction between the genes. We have the set of samples for which we have both the alterations and the target profile value. And now for each gene, we also know what is the subset of the samples in which it is mutated. or it is altered. We have a weight for each sample, and from the weights we also derived the penalty for each sample. And again, we are looking now for a connected subgraph of the input graph that has at most k genes and that maximizes the weight, where the weight is the same as we had before. So there is a component that relates to covering many samples with high value of the target, and the component that penalizes having non-mutually exclusive mutations. Okay, so again, one can prove just by a simple reduction from the problem considered by Uncover that this problem is NP-hard, so probably there is no polynomial time algorithm to solve all possible instances. And then what we did was actually to use an ILP formulation to solve the problem. And the ILP formulation is really the same formulation as Uncover, so there are the same variables, and there are the same constraints. Again, you can write down the objective function as this combination of covering high weight samples and penalizing non-mutually exclusive alterations. And then you wanted most k genes in your solution, you have your auxiliary constraints, and then there are additional constraints that correspond to some type of connectivity. And in particular, we look for high connectivity constraints, and by properly setting this value d here to be at least k/2, you obtain that what you have in output is both highly connected and you preserve the connectivity of the subgraph. Okay, so this is the way we are going to obtain a solution from our data, and then we want to also test the significance, the statistical significance of what we get. And for this, we use two permutation tests. The first one is the same as the test in Uncover, so you do a phenotype permutation, you permute the weights across the samples while everything else is fixed. And then the second test for significance is a network permutation where everything is fixed but the graph, and in a particular permutation you take a random graph that has the same node degree distribution as your input graph. So you preserve hubs and the degrees of every node, but now the specific associations between and the interactions between two different genes are actually destroyed and are going to be random. Okay, so what we did here was then first to use simulated data. Again, very similar to the one I described before, where you plant a solution in your data while other mutations are random. And we use these simulated data to both evaluate the ability of Netflix to identify the correct subnetwork and also to compare it with other methods, in particular with Uncover, and then also with the method from 2017, it's called Sigmod, that looks for subnetworks but really relies on single gene scores. So it doesn't really combine the different genes, but just look at an additive model for the sum of the genes in the subnetwork. Okay, so what we obtain is that all three methods are actually good in identifying the true subnetwork associated with the target profile. But then if you look at the false positive from the three methods, they are very different. In particular, Netflix has a low number of false positives, Uncover has a higher number of false positives, and now if you look at Sigmod, it has a very high number of false positives, since if you look here, the number of false positives, the average number is around 130. Okay, so Netflix, at least for simulated data, works well. And we then compared it with Uncover using real data from the GDSC drug sensitivity data from before. And what I'm showing here is a plot of the number of solutions reported by Uncover and reported by Netflix. And these are solutions for which the p-value from the permutation test is significant. And the different colors represent whether the solutions from Netflix and Uncover are actually the same, or they have two genes in common, or they have one gene in common, or they have no gene in common, and these are actually solutions that contains all genes that are never reported by the other method. So as we can see, Netflix and Uncover actually report fairly different solutions. And in this table, we have the different number of modules that are identified by the two methods. So for Uncover, it reports 156 significant modules, while Netflix finds more significant modules when you use the phenotype permutation testing. Then, if you consider also the network permutation testing, actually the number of modules that are reported as significant by Uncover decreases by a lot. So in particular, there are only 33 targets that have a significant solution when you use the permutation testing for the network. And this shows that actually the information in the network can be used to find modules that are probably more significant and more biologically significant than you would if you didn't consider the network. Okay, so now let me get just to some of the results that we have in the Netflix preprint that is available online. We have several interesting observations. Here I'm going to just describe one that I find particularly interesting. So we look at two different compounds and drugs, selumetinib and afatinib, and for both we look for modules, subnetworks that are associated either with decreased sensitivity of the drug or to increase sensitivity to the drug. And so these are the four different modules and the target profiles and the overall cover for the subnetwork. And an interesting observation here is that looking at these two targets, we identify subnetworks that are somehow similar, but they have an opposite association with the target for the two drugs. So for example, if you look at selumetinib with increased sensitivity, we get the BRAF and KRAS mutations, and KRAS and NRAS mutations are associated with decreased sensitivity to afatinib. And the similar results is what we observe for decreased sensitivity of selumetinib and increased sensitivity to afatinib. Okay? And this seems to suggest that actually these two drugs have a different and opposite type of sensitivity to the different types of alterations in the same pathway, in particular in the MAPK, RK, and AKT signaling pathway. And so it suggests that maybe a combination of the two drugs may work for a larger number of patients. And this is actually an interesting observation, I believe, also because there is a clinical trial for afatinib and selumetinib in combination that is currently underway for the non-small cell lung cancer. Okay, so these are the two main algorithms that I wanted to present, Uncover and Netflix. There are more results in the Netflix paper and also in the Uncover paper. And I just want to mention that we have also looked at other types of targets for which we can use our method, in particular in collaboration with Roded Sharan, Max Lizerson, and Teresa Prutskaya. We have looked at the use of Netflix to identify subnetworks of alterations that are associated with the mutational signatures that were discussed yesterday during the talks. And we have some interesting observations for those. And lastly, I want also to highlight the fact that we have developed methods to identify subnetworks that are associated with survival in cancer. And this is some work that started many years ago, looking at mutations associated with cancer, but only this year actually we published the journal version for finding the subnetworks associated with cancer. So to summarize, what I have presented today are, problems and algorithmic problems associated with finding this set of alterations that have a functional association with the phenotypes, that could be clinical phenotypes as well. And the two main algorithms we have developed, Uncover and Netflix, for this problem. There are many challenges that I would be happy to discuss in getting these methods to improve even further on data. So I want to now acknowledge the funding sources and the main people that were involved in this project. So Uncover was driven mostly by Rebecca Sartobasso, that is a PhD student in Berkeley, in Dorit Oakbaum lab, while Netflix is driven both by Rebecca and Yuan Kim, that is part of the lab of Teresa Prutskaya at the NIH. The part on the signatures in collaboration with all these people I just mentioned, and also Max Lizerson and Rodel Sharan and students in their lab, while the work on survival analysis is mostly done by a postdoc in my lab, Federico Altieri, and also a master student, Tommy Hansen, that worked with me when I was at SDU in Denmark. So I just now want to steal one more minute of your time for a shameless advertising that hopefully is useful for everybody. And in particular, we're going to organize Recom 2020 in Padua, in Italy, so it's going to be May 10 to 13. I hope these pictures are enough to convince you to come to Padua. If Recom by itself was not enough, I'm sure Recom by itself is more than enough, but in case you needed some more proofs of, or more evidence of why it would be a good idea to get there, these are some pictures. I'm Italian, so I can also testify on the great quality of food that you will find there. Okay. So I look forward to see all of you there, and please submit your best work to Recom. And thank you, and if you have any questions, I would be happy to answer.